and get started. I'm very happy to introduce one of our own local students, Cory Scott, who is a sixth-year PhD student here at UCI. So before coming to UCI, Cory earned a bachelor's degree in math and computer science from Colorado College. And he's interested in a lot of different problems. He works on in multiscale modeling and machine learning with his advisor, Professor Eric Multiscale. And today in particular, he's going to be talking about modeling of microtubule energy potentials with graph prolongation convolutional networks. Cory, please tell us about it.
Thanks, Eric. So, like Eric said, this is work with my PhD advisor, also named Eric Multiscale. And basically what I'm going to talk about today is a little bit about a simulation that I wrote, a very coarse-grained simulation of a microtubule. If you don't know what a microtubule is, I'll get into that in just a second. And then I'm going to talk about trying to recreate the dynamics of that simulation with a new type of machine learning model that Professor Multiscale and I invented. And then I'm going to wrap up by talking about some future directions for this work, which is basically extending these machine learning models, which operate on graphs, to objects that are in some sense the continuum limit of graphs embedded in three-dimensional Euclidean space, which are called cell complexes. First, I want to start off by giving a couple of references. So if you are not familiar with graph convolutional networks or graph neural networks in general, this is a review paper from a little under a year ago that I'm pretty fond of. I think it's a very broad review that presents things in a fairly unified way, so I would recommend that. And then secondly, the other link there is a link to the recently published paper on the work that I'm going to be presenting today, if you'd like any more detail about anything that I discuss. So first, I want to talk about what is a microtubule, if you don't know. So microtubules are basically organic nanotubes that are repeating structures made out of this protein called tubulin. And tubulin comes in two varieties, alpha tubulin and beta tubulin, and those two associate into these alpha-beta dimers, and then dimers have a preferential association into strands, which then like to associate side by side into tubes, and these tubes, microtubules, have a lot of interesting properties, and they play a bunch of interesting roles in the cell. So they're generally about 25, 24 nanometers in diameter. They can be up to 50 micrometers long, and they play a lot of interesting roles in the dynamic cytoskeleton of the cell. And those roles, that interesting behavior, comes about as a result of a bunch of the small-scale dynamic properties of microtubules. So that's what we're going to try to talk about. And then this video, which played a second ago and which I'll play again, is a video of a microscope view of a bunch of microtubules moving around in a cell from a paper in cell biology. So like I said, microtubules have some very interesting properties, one of which is that they grow. So once a microtubule starts to form, then it tends to associate free-floating tubulin that's in its environment to lengthen itself. And those free-floating tubulin dimers kind of click into place in the lattice of the microtubule. And I say lattice because microtubules are this sort of helical repeating structure of those alpha-beta subunits. There's generally 13 or so strands in a microtubule, and then those strands are in a lattice, a coiling lattice that goes around. So microtubules grow by associating tubulin at one end, but they also exhibit this interesting property called dynamic instability, where they can enter what's called a catastrophe phase. And in the catastrophe phase, they sort of suddenly unravel and dissolve. But they can rescue from catastrophe and start growing again. And so you get this interesting behavior of growth, catastrophe, rescue, more growth, etc. They can also bundle. So this is another microscope view of a couple of microtubules basically colliding at a very slight angle and infusing together, or wrapping around one another. And so microtubules, by bundling, can form these really complex, really strong, rigid structures inside the cell. And then finally, the property that I'm going to be talking the most about today is their stiffness. So because they're this nanotube structure, microtubules are very stiff to any kind of bending motion. So to get into that a little bit more, this is a close-up view of the structure that I used in all of my simulations, and I'll explain more about that in a moment. But the basic idea is that that you can see in this view that if you imagine each tubulin monomer as a little bead connected to its neighbors by springs that are trying to hold it in place in this lattice, that when you try to bend that whole structure, all of those associations, all of those constraints are being violated. And so if you add up the degree to which every constraint gets violated and consider that a kind of potential energy term, then you get this sort of heat map of how much each tubulin monomer considers its local constraints to be violated. What do I mean by constraints? I'll get into that in a second. So, like I said, microtubules are very stiff. They have a Young's modulus of 4.6 by 10 to the 8th newtons per meter squared. For reference, that's about the same as plexiglass. Granted, these are wildly different length scales, right? But you can sort of think of them as these little plexiglass cylinders that are inside the cell and help do a lot of different mechanical things inside the cell. So this is a simulation that I built using the molecular dynamics simulation tool LAMMPS, which is a distributed molecular dynamics code that's put out by Sandia National Labs. And LAMMPS basically allows you to define a bunch of those energetic interactions that I referred to a moment ago by defining types of particles and defining the interactions between particles. And then you can tell LAMMPS, okay, I want to put these external conditions on my protein or my molecule, and I want to run for this many time steps where each time step represents some unit of real time. And so in this case, what the actual simulation that I'm running is the microtubule segment is clamped at one end, and then the other end is being pulled down with a force load. And that force load starts out small and ramps up to a constant value. And so by the end of the simulation, the whole structure has reached kind of an equilibrium where the springiness of the microtubule is pushing back against the force that's trying to pull one side of it down. So I talked a second ago about association energies, and those take the form of a bunch of rules that are written out for interactions between tubulin monomers. So for example, we can say that any pair of tubulin monomers that are associated have to be a specific distance apart, right? If they move further apart or closer together, there's a little spring there that's keeping them that distance apart. But we can also talk about angle constraints. So there's sort of a little angular spring that holds triplets of tubulin monomers to try to keep them from doing this or from expanding further. And the equations for those are what I have on the slide here, right? So we have some strength parameter, that's L type B and L type theta there. That's just a constant, and that basically says how strong each association is. And then we have a term that basically says the length of the bond should be this, or else the violation, the energy, is going to increase with a squared term multiplied by this constant according to how far the actual length of that bond is away from what we think the actual bond length should be. And there's lots of different arrangements of alpha and beta tubulin within that whole structure. And so part of the work that I had to do to get the simulation working was going through and tabulating all the different ways, for example, for the triplets, you can have an alpha alpha beta, beta alpha beta, all the possible combinations, but then that triplet can be in different orientations with respect to the long axis of the microtubule. And so I basically had to tabulate all of those and define a different energy term for each of them. So we're going to use that simulation to generate a data set. And what I mean by that is that we're going to sweep over different strength parameters for all of those interaction terms that I mentioned a second ago. So there's 12 or so parameters, and we're going to sweep over lots of different values for each. Yeah, there's the parameters. I guess there's 7 there. And those are in units of energy over distance squared. And so for each combination of parameters, we're going to simulate them for a set amount of time. And like I said before, the force that's pulling on the end is going to slowly ramp up to a constant force. And then we're going to continue simulating until the system kind of reaches equilibrium. And every so often, we're going to save all of the positions of every monomer, the velocity of every monomer, and the energy that all of those rules we defined predicts for every monomer. So for every time step, for every combination of parameters, we're getting a position for each monomer and a velocity for each monomer, and then our output is going to be the energy per monomer. I want to, in the interest of full disclosure, talk about a couple caveats of this simulation method. Like I said, the input to our machine learning model is going to be that position and velocity per monomer. And that the output is going to be trying to recreate the energy potentials that the deterministic molecular dynamics simulation code produces. But there's some caveats to this simulation. The first is that the simulation isn't actually at room temperature. The simulated temperature is much colder, and that was because it was difficult to get a microtubule model that was mechanically stable when it experienced sort of the random noise, the random molecular fluctuations resulting from being in a room temperature solvent. Other than the final Young's modulus number, there isn't a lot of ground truth to compare to. There's not a lot of work about the dynamics of microtubules considered at this exact scale. There's lots of work modeling tubulin monomers by themselves. There's lots of work modeling clusters of microtubules, but this sort of treatment of each tubulin monomer as being a single bead, there's not a lot of ground truth for us to compare the simulation to. So the simulation could definitely be refined further, but for now, we're going to take the simulation output as our ground truth and use it to see how our multiscale machine learning method does. And just a final caveat, the graph size here, we have a fixed microtubule graph. And what that means is that we don't have access to some of the interesting behaviors that I detailed on the slide a couple seconds ago, like dynamic instability or bundling. Those require you to be able to change the size of your graph, which we can't do yet, but we hope to in the future. Really quick for anyone who hasn't seen it, because this is a machine learning talk, I'm not going to spend too much time talking about the details here, but a lot of people are familiar with regular image or video convolution, where you have a regular grid and you're sliding some kind of trained filters over that grid to try to classify whether something's a cat or a dog, or identify a pedestrian in a scene. So that's generally just called convolution or convolutional neural networks. But there's a corresponding approach where your underlying structure is not a regular grid, it's an arbitrary graph. And that's called graph convolution. But the basic ideas are the same. The basic ideas are we're going to take each node and look in a certain local neighborhood and then use a nonlinear function to update that node's state. And we're going to stack up several layers of that. And then at the end, we're going to predict something, be it label or be it potential energy or be it positions for some embedding of the graph, etc. So the machine learning model that we propose is a multi-scale version of graph convolution. There's sort of a cartoon of what our model does on this slide. So the x sub I are inputs, and you can see that we sort of incrementally downsample each of the xi's until a certain coarsest scale, and then each of those inputs, which are at different spatial scales, gets fed into its own graph convolutional network. And then when we get a set of outputs, we're going to upsample all of them using the transpose of the same downsampling operator. And once we have all of the upsampled outputs, then we just add them together, and that's the final output of the network. For anyone who has worked in any kind of segmentation, image segmentation context, this probably looks a lot similar to UNet, which is I think a 2012 paper that used this same kind of multiscale approach for segmenting of medical images. And the basic idea is that you have a path in your network that downsamples and then upsamples, but then you have these skip connections that stay at the same scale. And so we're doing the same type of thing, but the sort of secret ingredient is our downsampling and upsampling operators. And I'll get into how those are produced in a moment. Here's just a slightly larger view of the bottom of that cartoon showing that if we're at a specific scale, we have an input that comes in from the left. We downsample it by multiplying it with a projection matrix, and then it gets fed into the machine learning model at that scale. And then, after the output, we have another model output that comes in from from below. The output at the current level gets added, and then we again upsample by multiplying with p sub j minus 1. So really the takeaway of these two diagrams is that we downsample with a projection matrix, just a matrix multiplication, and then we upsample by again multiplying with the same matrix, but now it's the matrix, not the transpose of the matrix as it is in the downsampling case. So that's all well and good, but I've sort of under the rug how we find a good coarsened representation of our graph, right? So I have this microtubule graph, and I have a machine learning model that operates at multiple spatial scales, but the microtubule graph is only the finest scale of all of the graphs that I'll use in my model. I don't necessarily know a priori what good coarsening, what good coarser models might, or coarser graphs might be a good fit for my task. So in order to find coarsened representations in a systematic way, we use something called graph diffusion distance. And the basic idea is that we want to make sure that those P matrices, those projection matrices, optimally align the Laplacians of the two graphs. The Laplacian is just a matrix that depends on the adjacency matrix of the graph, and you can think of it as describing what diffusion looks like on the nodes of the graph. If I were letting little particles of ink randomly jump around the graph, the Laplacian sort of describes what their behavior is going to look like. And so if we have the idea is that if we have a P matrix that successfully makes the dynamics of diffusion on graph A and B look similar, then we might be in the running to get other dynamics on those two graphs to look similar to one another. And so we're going to find a coarsened representation that has a small graph diffusion distance from our original graph. And so this is what that looks like. the different, I guess, different axes of this table don't matter so much. The basic idea is that we have a way to generate lots of proposal sort of extruded tube-like graphs, and we're going to take as our coarse representation the one that has the smallest graph diffusion distance to our original microtubule graph. And so we generate a bunch of these extruded tube-like graphs, and then the one that we pick as our coarse scale representation is the one that is closest to the fine scale representation from the point of view of diffusion on the nodes of the graph. And so when we do that with our microtubule graph, and we decide just totally arbitrarily to do it with 3 different scales, these are the 3 graphs we get. So you can sort of see that there's the fine scale, the original microtubule, there's an intermediate scale, and then there's the coarsest scale. And if you look, the intermediate scale is sort of almost exactly what we would expect as sort of a first order coarsening of our original microtubule, because what we have there is each alpha beta tubulin pair has been coalesced into a single node in the intermediate scale, right? So we've gone from treating each tubulin as a node to treating each alpha beta dimer as a node. And then the coarsest scale doesn't necessarily have a clear interpretation in terms of tubulin monomers anymore, but the point is really just that we have this hierarchical representation of our original graph, of successive coarsenings of our original graph. So the basic idea behind this kind of multiscale training method is that graph convolution, each layer of graph convolution has computational cost that is big O of the number of nodes in the graph times the number of edges in the graph times the number of channels. So the amount of information you have at each node. And so what this approach lets us do is it lets us have many, many more channels, much more information per node at the coarsest scale, right? Because we've cut down the size of the graph, we've cut down both the number of nodes and number of edges by a lot, which means that we can have more filters, more features per node at the coarsest scale for the same computational cost. And so one way you could think of this is that we're taking what would be a very complex mapping from input to output, and we're sort of taking the most complex part of that decision and putting it down at the coarsest scale. And then the intermediate scale has to make that decision for more nodes, but it's a simpler decision because it's informed by information from the coarse scale. And then that process repeats for every scale that you have. And just as a side note, when we are training this structure, we can make a choice about whether we let the error gradients change the projection matrices or not. So the projection matrices are initialized as being the ones that achieved the best diffusion distance in our diffusion distance optimization. But then once we create a machine learning model with those P matrices as our projection operators, we can decide to either leave them fixed during training, or we can let them vary according to the gradients that the network as a whole is receiving from its error. Because it's easy to just pass gradients through matrix multiplication. So again, because this is technically a graph convolutional network ensemble, it's not really fair to just compare to a single graph convolutional network. So what we compare to are different ensemble methods. So the first method is we can compare to an ensemble that just has a single GCN, an ensemble that has 2 GCNs, an ensemble that has 3 GCNs, all of which are operating at the fine scale with as many filters as we have in our multiscale network. And so those are going to be much more expensive, but they have sort of the same chance of figuring out the mapping function that the finest scale in our hierarchy is figuring out. And then there's another multiscale ensemble method called NGCN, which I won't get into the details for lack of time, but that approach basically aggregates information from larger and larger areas in the graph. So it's doing a very similar thing, but at larger computational cost. So what we see in this chart is that our method's validation error decreases much faster as a function of how many flops we spend training the model. So this is on a log-log axis, and the adaptive version of our model with 3 layers in it trains much more quickly and efficiently than any of the other ensemble methods we considered, and it ends up doing better in terms of error by basically a whole order of magnitude. So for me, this sort of just shows the inherent power of multiscale oriented methods in machine learning. But another thing that we can always do when we have this kind of hierarchical multiscale method is that we can adapt our training procedure to take advantage of the fact that things are multiscale. So this is adapted from a collection of techniques called multigrid methods. Again, those are sort of beyond the scope of this talk, but the idea is that anytime you have this hierarchical representation of some problem, you can spend more time working at the coarse scale, and that if you have a really good coarse scale solution, that's a good place for a fine scale solution to start from. And so we can adapt our training schedule to say, train the finest level in our hierarchy, and then the intermediate one, and then the coarsest, and then back to intermediate, back to fine. That would be gamma equals 1. Gamma is just a recursion parameter that says how often you recurse to a coarser level in the hierarchy. But we can also, make 2 stops to the coarser levels of the hierarchy for every round of training we do at the finest scale. So basically, the takeaway from this plot is that we can train the models that are at different levels in the hierarchy at different times in our entire run of training. And when we do that, we get a further increase. So again, on this plot, I have a comparison to a single GCN model, and then I have several different methods of moving through training schedules for the whole hierarchy top to bottom. So for example, you can imagine starting out training only the coarsest model, and then training the coarse model and the intermediate model, and then training the coarse model, intermediate model, and fine model all at the same time. And that's the sort of purplish dashed line. But what ends up sort of outperforming everything else is the W cycle schedule. So that's basically the gamma equals 2 method here, where you start out at the finest scale, you go down to the coarsest scale, and then you sort of do these little hiccups up until eventually you get back to the finest scale again later. And so that ends up outperforming everything else, and indeed it even outperforms just having the multiscale hierarchy on its own. So basically the multiscale hierarchy on its own did fairly well, but then with this alternate training schedule, we do even better. So one sort of quick note is this idea of having learned projection operators isn't necessarily new to us. There's been a lot of work on how to dynamically coarsen graphs in graph neural networks. But what we see is that those coarsening methods are really expensive. So the sort of gold standard of those methods, at least right now, is something called differentiable pooling. And differentiable pooling has basically an auxiliary network at each layer of the graph neural network. There's a second a second graph neural network whose job it is to predict the coarsening operator for the next layer. And that means that you basically have twice as many graph neural network layers. They're not all, it's not a direct 2 times computational cost because those layers are all going to be different numbers of filters and different sizes. But the point is that works, but it is pretty expensive in terms of the amount of training that you have to do. And so when we compare our method to the differentiable pooling method, we still come out ahead, even though in theory the differentiable pooling method should be able to find the same reasonably good projection operators between scales that we start out with. When we initialize them with these projection matrices that are optimized to be the best matrices for diffusion distance, we get this speed up right out of the gate. We don't need an auxiliary neural network to predict the projection maps. And so here are the results for all the methods that I've mentioned. So you can see that like I said on the main slide, the adaptive graph prolongation convolutional network does better than any of the other methods by an order of magnitude, more or less. And these numbers show mean and standard deviation that was calculated by running several trials with different random seeds and collecting the validation error for each. So these are different validation sets. Really briefly, I want to talk about a bonus that you get by using a neural network model to do this kind of molecular dynamics. So a really interesting set of developments in the last couple of years has been the use of machine learning models, specifically because they're differentiable, to do things like optimize bridge structures and solve PDEs and lots of other different sort of physics-like, simulation-like approaches where you want to approximate some simulation with your neural network model, and once you do that, the neural network model is an approximator, but it's also differentiable. And so you can do things like you can collect the derivative of the output with respect to the input. And the reason that's relevant in the molecular dynamics context is that our inputs are positions and our outputs are energies. And so the derivative of energy with respect to position is a force. And what that lets us do is it lets us get forces acting on all of the molecules and lets us then directly simulate molecular dynamics using the trained neural network. So the idea is that you could take a position of your molecule, push that through the network and get the energy out, collect all the gradients and find the gradient of the inputs with respect to position, and then that lets you move each particle in your simulation by some amount to minimize that energy. But you don't just have to do it with the position outputs. You could also do it with any other parameters that you might have. So in this experiment, each input, the neural network got to know what the force constants were, the constants out at the front of each of those spring energy laws. And you can imagine if we had some ground truth, then we could basically feed that ground truth position information into the network and use the output to use the gradient to find what set of bond association strengths gives us behavior that's similar to what our ground truth is. So that's a lot of information, but the basic idea is you get a lot of interesting behavior for free when you use a differentiable model to approximate your molecular dynamics simulator. So I'm going to kind of totally pivot and talk about a different project that I hope to dovetail together with this one. And the common thread in both of these is graph diffusion distance. And so what I'm going to talk about for a moment is using graph diffusion distance to try and produce fixed size representations of cell complexes in three-dimensional Euclidean space. Doesn't necessarily have to be 3 dimensions. That's just the examples that I've been working with right now. You see a couple of them on this slide. So there's a sort of a long tube, but there's this kind of barbell shape. There's a torus. And for anyone that isn't familiar with the term, a cell complex is basically a bunch of manifolds, which you can think of as lower dimensional objects in R3. A cell complex is a bunch of those, along with information about how they're glued together. So just as an example, here's a shape that is not a manifold, right? It's sort of a pork pie hat shape with one cylinder and then another cylinder at its equator, more or less. And this isn't just a manifold. It's a more complex object. It's a cell complex. And so the idea is to try to use graph diffusion distance to produce a representation of these kinds of objects that doesn't depend on how many nodes you have. That's sort of dataset size invariant. So here I've just evenly spread a bunch of points randomly over the surface of my pork pie hat object. But you can imagine wanting to get some kind of representation that doesn't necessarily depend on whether you've scattered 1,000 points or 1,600 or 10,000 points on the surface of your cell complex. So the way that we do that is we take our graph that we get by discretizing that space, and we cut it up into little local neighborhoods. And you can do that using whatever graph clustering scheme is your favorite. Right now, I think that this uses, I think what I'm using to produce this plot is affinity propagation, but again, that doesn't necessarily matter. What matters is that we're cutting the graph up into all these little local chunks. And then what we do is we want to calculate the graph diffusion distance between those patches. So we're taking those patches and we're comparing them in terms of what diffusion looks like locally for each patch. And so in the examples that are at the bottom of the slide, we have the red patch is the same in both images, and you can see that the red patch kind of takes up a little bit of the equator. And so when we compare that to the yellow patch, which also takes up a little bit of the equator, versus a patch that's way out on the rim, those are going to look different from the point of view of ink diffusing randomly over the nodes. And so the idea is that we're sort of taking these local patches and comparing them to one another in terms of whether diffusion is similar in those areas. And when we do that, we can get a new representation of the graph where we've coarsened the graph according to our coarsening algorithm, and we've put down weighted edges according to how similar neighboring patches are in terms of that diffusion distance operation. I know that the slide is throwing a lot at you, but the basic idea is if we do that and then we compare it to directly doing the graph diffusion distance between the two graphs in question, we end up getting a representation or a distance that is much more robust than just graph diffusion distance or other diffusion or other graph distance metrics. So when we coarsen two of these cell complexes that way, and then we compute the diffusion distance between the coarsened versions, then we end up being able to, those distances lead to much tighter clusters. And so you can see at the bottom, compared to other ways of comparing these objects, this method of comparing local neighborhoods to each other and building this coarsened graph has 100% k-means accuracy over a small data set that I constructed that has a bunch of the objects that you've already seen, basically. So the idea is I have a bunch of these 3D objects, I coarsen them all, I compare all the coarsened representations, and the coarsening method ends up giving us tighter clusters than other graph comparisons between those objects. So the hope is that this gives us a fixed size, because again, we can pick the number of communities we divide the graph into, a fixed size representation of the cell complex that we can then feed into a later machine learning model, right? And because it's fixed size, we can, because it's fixed size, we avoid a lot of the drawbacks typically associated with graph convolution, like needing to use sparse matrix multiplication and things like that. And this is just a zoom in. You can see that we have pretty good separation of all the different objects in the little zoo of cell complexes that I made. And I should say this is really an initial effort at this. There's some data sets that exist of 3D objects to recognize, and that's kind of the next step for this branch of my work, is applying it to one of those more real data sets. But to tie it back to microtubules, one aspect of graph diffusion distance is that as the graphs get very, very large or very, very dense, there's this kind of limiting behavior of graph diffusion distance. And so as the graphs get larger and larger, eventually your optimal projection operator converges to some limit projection operator. I'm not going to define any of those terms technically. And the hope is that we can apply that to molecular dynamics, basically by shrink wrapping the whole molecule and using using that exterior shell of each molecule as our cell complex. And that's the sort of shrink wrapped molecule approach is something that's been done in a few molecular dynamics papers for predicting things like docking, affinity, and related functions of the surface of a molecule. And so the hope would be that we can maybe do this for a model of a microtubule, and that would be sort of one step finer than the finest scale that I presented so far. Because if you go from having your tubulin monomers be these little beads to then being, these more complex cell manifold type shapes, then you can model more interesting things happening, like the tubulin monomers being squished or deformed or things like that. So just as a summary, what I've talked about today is constructing a synthetic data set of a protein molecule under mechanical load. I've shown that a machine learning model that Professor Mulesness and I have developed is much more efficient applied to the problem of predicting the energetic dynamics of that protein in our simulation. And I hope that I've shown the initial steps of taking that idea and generalizing it to really, really large graphs, things that are sort of the continuum limit of a sequence of finer and finer discretizations of a cell complex. And so for future work, I would like to make the simulation a little bit more true to life, what microtubules do in real life. So for example, bringing it up to room temperature, having more complicated potentials. I would like to be able to handle bundling and association, but those are vastly more complicated. The other thing is that a current reigning hypothesis is that microtubules buckle in places where they're weak, and that missing monomers or defects in that lattice might be responsible. And so the hope would be that we can model those dynamics as well. And speaking of dynamics, really, in order to get there, this model has to be extended to handle dynamic graphs. So not just static, this is my microtubule model, this is my coarsened microtubule model, but some way of growing the model over time. And again, that would let us model the really interesting association to the free end and catastrophe and rescue kind of behavior that is responsible for a lot of the more interesting microtubule dynamics. And then finally, extending graph diffusion distance to the sort of infinite limit objects that I was talking about a second ago, cell complexes, and building fixed size representations of those objects. Another point under that I didn't mention is that the hope would be that if you have a distance measure between such things, that you could train your machine learning model and then investigate what manifold the machine learning model has learned by using these diffusion distance methods. So at this point, I just have a limited set of the references that I've used up on this slide. I'd like to thank my funding sources, so National Institute of Aging grant, an HSFP grant, and a National Science Foundation NRT award. And actually, our own department's Porrick Smith is the PI of that award. So thank you to all those funding sources, and I'm happy to take questions at this time.
Okay, great. Thank you, Cory. So if anyone has questions, you can either sort of unmute and ask them, or also type a question into the chat window. I'll go ahead and ask. I actually had a couple questions about the first half, the kind of microtubule deformation. So I think you had some comparisons of kind of different graph neural network and ensembles and things, but what I missed, I think, is so how does your sort of, once you've learned this graph neural network predictor of energy, how does that compare to the complexity of this original simulator that you used?
I see. Yeah. So the sort of bitter lesson, to steal that term, is that a machine learning model is basically never going to be as fast as a extremely optimized, extremely parallelized molecular dynamics code. So we're not necessarily, we're not trying to compete with those codes in terms of speed for a fixed size time step. But the hope would be that if you have a machine learning model that's well trained enough, you could take larger time steps. And if you're taking larger time steps, then you need to do fewer of them. And so then you might end up still getting out ahead, right? If a machine learning model time step takes 1,000 times longer, and that's not the actual number, I'm just throwing that out. If it takes 1,000 times longer, but you need to do, you need to do 10 of them for every 1,000, every 1,000 time steps that you would do with the, so I got caught up in the numbers. The point is that we can accelerate these simulations by taking time steps that represent more time within the simulation than the deterministic model can do. That's the eventual hope here.
And did you think about like testing generalization in this context? Like you could imagine, take a slightly different microtubule structure, give it some different deformations, and say, can my network still predict those, right? Or have you looked at that? And if you haven't, do you have hypotheses on how far would generalize versus how brittle it would be?
So I don't necessarily have a sense of that, but that is on the long list of places to take this project. And in particular, that when I mentioned on a slide a second ago about missing monomers or defects in the lattice, that's in order to cover that case, I think what you would need to do is you would need to have a really general model so that you could, delete an arbitrary monomer out of the lattice and not have to train your model on every single possible way that there could be defects in your microtubule lattice, but hopefully train a model that recreates the energetic dynamics generally enough that you can delete monomers and still have an okay answer at the end. As far as actual ways of doing that, I have variants of this simulation coded up, things like other mechanical deformations, like taking both ends and twisting them, or just taking both ends and trying to pull it this way rather than bending it. Those were not in the data set for any of the training that I've talked about so far. But I guess my hope would be that if you had a data set that had enough of those types of deformations in it, that you would get pretty good coverage in terms of generalizing to other positions.
Okay, yeah, that makes sense. Are there other questions for Cory?
Yeah, great talk. This could have really nice applications in control and reinforcement, as you can imagine. What I'm curious, and sorry if I missed it, is so you have a nice method for measuring this diffusion distance between graphs, different coarsenings of the graph, but how do you generate candidate coarsenings?
So in this case, that was fairly easy because we knew that the candidates all needed to be tubes, right? Extruded tubes of some kind, in order to model the bending behavior that we wanted to have in the final model. So in this case, what I did was I searched over all possible numbers of filaments. So that's the filament is kind of the longitudinal strand of tubulin monomers, and the finest scale has 13 of those in a spiral, right? So to generate the coarsened model, in this case, I just looked at tubes with varying helical offsets and varying numbers of protofilaments, and I took the one of those that had the smallest diffusion distance to the original graph. If you didn't have a good hypothesis, then that would have to be another kind of layer of optimization, is coming up with a hypothesis and then computing the graph diffusion distance to see whether it's a good coarsening under that measure.
Yeah, makes sense. That's often the challenge in hierarchical reinforcement learning, but your distance measure can still be really nice addition. Thank you.
Thank you.
Thank you. Any other questions? Okay, well, I'm sure we're all virtually thanking Cory for a great talk. Thank you all for joining us, and we'll continue to be back in the coming weeks for more CML seminars. Cheers.
Thanks, everyone.